Hello, my name is Campbell Elam, and today I'm going to be talking about the immunological perspective of type 1 diabetes. Type 1 diabetes mellitus is an autoimmune disorder involving the destruction of the beta cells of the pancreas, which produce insulin. This therefore results in an impaired ability to metabolize glucose. Therefore, affected individuals will require lifelong insulin replacement, and there's no known cure for this illness. Still, the etiology and pathophysiology of type 1 diabetes is largely unknown. Despite the relatively unknown etiology of the disease, type 1 diabetes is still heavily researched as it affects over 1 million Americans. The first stage of type 1 diabetes progression is mainly asymptomatic and appears in the early years of childhood before beta cells are fully destructed. It's marked by normal glucose metabolism and a minimum of 2 pancreatic autoantibodies present. In the second stage of the disease, the pancreatic autoantibodies are present and the destruction of beta cells continues. It's marked by asymptomatic dysglycemia, meaning that the beta cells are not producing normal insulin, and the body continues to attack beta cells as a full immune response is launched onto the pancreas. In the final stage of disease progression, type 1 diabetes has fully manifested. Beta cell destruction has reached a critical mass, and the symptoms are fully present. In order to understand the complexity of this autoimmune reaction, we need to start with the innate immune system. The first line of defense in the body against an infection is the innate immune system, which is consequently thought to be the first event in the manifestation of type 1 diabetes. One major and well-known receptor within the innate immune system is the toll-like receptor, which, upon detection of an antigen from a pathogen, will cause a signaling cascade that activates the immune system, leading to the production of inflammatory molecules like Th1 cytokines and the additional coordination of the adaptive immune system mechanisms against infections. This is thought to play a role in the manifestation of type 1 diabetes as increased TLR concentrations and signaling cascades are observed within the islets of type 1 diabetes patients. Subsequent inflammation in the islets of the pancreas have been shown to damage beta cells, leading to the progressive loss of insulin production. It furthermore has been known to set off a sequence of events involving more immune cells being recruited at certain times within the innate immune system malfunction. The initial cause of this malfunction of the innate immune system is unknown, but viral infections have been associated with the development of type 1 diabetes in stage 1 patients in conjugation with autoreactive T and B cells. For this reason, it is believed that the beta cells themselves play a role in their own destruction, as malfunctioning inflammation and cytokine signaling can recruit other cells, resulting in further immune attack and destruction of the beta cells in the pancreas. Despite the clear links between the innate immune system malfunctioning and type 1 diabetes progression, it's not the only thing involved in this disease. B cells play a major role in type 1 diabetes disease pathophysiology. As many as 70% of B cells produced are reactive to self-antigens. These self-reacting B cells are normally cold, but in type 1 diabetic patients, B cells reactive to self-islet cells proliferate and escape negative selection through an unknown mechanism. In junction with a primed innate response, this leads to the development of type 1 diabetes mellitus. These autoreactive B cells, often attracted and activated by underlying innate inflammation responses and cytokine signaling from the beta cells themselves, are integral to the autoimmune response. Collaboration with CD4 cells assists B cell differentiation to produce self-reactive cytokines and plasma cells to start producing autoantibodies. These immune complexes are thought to invade the glomeruli of the pancreas, leading to the thickening of the glomerular basement membrane and subsequent inflammation. These, in turn, produce a greater cascade effect, as broken down beta cells produce DAMPs and a further autoimmune response within B and T cells. To solidify this connection, a clinical trial found that B cell depletion therapy, in which B cells are reduced in type 1 diabetic patients, proved some efficacy in treating type 1 diabetes. The innate immune system and B cell autoreactivity isn't enough to completely kill beta cells. They need T cells in order to actually provide cytotoxic effects and completely manifest the disease. The activity of islet reactive T cells are currently the strongest and most studied link we have in tracing the physiopathology of type 1 diabetes manifestation. CD4 and CD8 cells in type 1 diabetic patients, like B cells, undergo negative selection escaping and will react with B cells. Those B cells additionally activate CD8 cells to further attack beta cells through the release of porphyrin and granzyme B with the help of antigen presenting dendritic cells. CD4 cells are recruited by the cross presentation of beta cell antigens to help through the release of pro inflammatory IFN22. Type 1 diabetic islets furthermore display extremely high levels of self reactive T cell activity, which additionally has been proven to be cytotoxic to B cells in vitro. In short, T cells are the main direct cause of beta cell death, activated by autoreactive B cells and attracted by a malfunctioning innate immune response. Type 1 diabetes is a massively complex autoimmune disorder whose etiology and pathophysiology is not well understood. However, the progression of type 1 diabetes seems to begin within the innate immune system with abnormal inflammation and cytokine signaling seemingly caused by the beta cells themselves. It is possible that an early viral infection increases the risk of the disease development. This attracts the activity of autoreactive B cells, which present self antigens to other immune cells and directly cause some damaging inflammation themselves. Finally, in conjugation with those steps, cytotoxic CD8 cells actively kill beta cells after activation by autoreactive B and dendritic cells. They are the most clear link between the immune system and disease development. Although the exact mechanisms behind these malfunctions is unclear, it is apparent that type 1 diabetes is the result of multiple self reactive events and multiple facets of the immune system, resulting in a kind of perfect storm. This may explain why the cause of type 1 diabetes can't be traced to one specific event, such as a genetic mutation.